Thanks for coming on, Doug. I think right off the bat, we can jump right into setting the stage and kind of answer the question, what is Plex?
Sure. Yeah, so Plex Research is the company I founded about 8 years ago now. And it really all came out of this idea that you had this really successful algorithm that Google invented, PageRank, this idea that you can take the internet and you can use centrality algorithms and you can figure out where is everyone pointing to, what are all the web pages linking to, and then that's a way to decide what's relevant. And the idea is that you can take that sort of approach, but instead of web pages, you can apply it to data. So this is something that can be used actually, in our case, with biomedical data. And you could take massive amounts of even relatively raw data and ask, where does it converge? Does it point to some answer for a given question? And so that's sort of the core idea that Plex was founded around, using different kinds of centrality algorithms to look for answers in all of the data that we've collected.
Got it. And what is the reason all this data is separated? Why is it compartmentalized? Why is it not together? And how can we go about getting ready with Plex to collect all that data?
Yeah, I mean, there's been this long-standing issue within data science and in biology and in drug discovery that we collect all kinds of data, really diverse data, large-scale data, but it ends up in silos because of the diversity of the data, the complexity, all of these reasons. And so it's spread all over. And we have this publication process that just hasn't kept up with technology. So that's the other thing I really leverage, we really exploit is, certainly the idea of centrality, but the reason it's such a huge opportunity in the space is because we have just incredible amounts of data that we can't really access. So we have this publication process where there's, it's based on text. We have, you generate your data, you write your paper, there's around 5,000 words, give or take. And then millions of data points get deposited in the supplemental file, sometimes never to be seen again. Right. And there's maybe thousands of these. There was a recent survey that found there were 6,000 biology databases, right? Just databases.
Yep.
And meanwhile, we have all of these questions that come up. We're working on various diseases and different approaches. And we'd like to know, for this disease or this thing we're working on, what does all of that data have to say about it, right? And the vast majority of the data in all of those studies was not captured in the paper. If it was, published in some peer-reviewed journal or in whatever form, the vast majority of the data they collected didn't find its way into the text of the paper or even into the figures, sort of sitting in all these tables. So, there hasn't been an efficient way to bring it all together and say, okay, we looked at all the world's data, and here's what it has to say. Like, it points to this, this answer. And so it's really the combination of those things that really makes Plex so powerful. It's that insight about let's apply, centrality algorithms, and PageRank is one of mankind's most famous and successful algorithms. It's the reason that Google is Google, right? So taking that kind of algorithm and then combining it with the insight or the issue we have of data silos to allow usage of just massive amounts of the raw data, all the data that didn't make it into the text, to answer questions, right? So because we actually do have the answers to many questions, in those data, and we just don't realize it.
Yeah, great high level. We'll dive more into the nuance of the product and the science that's being done by the research teams and all that. But I think that's a good starting point. Now let's set the stage on what brought you to actually start Plex. So if you'd go over your early career and PhD and all that stuff, that'd be awesome.
Yeah, so I did my PhD at Harvard in George Church's lab. I was there back when microarrays were brand new, so I published some of the very first papers on microarrays. And this is a technology for looking at the activity of all the genes, large-scale gene activity. And it was sort of the beginning of what they call functional genomics, where we're looking at the functions of all the genes and how they interact and behave. So published some of the first papers in that space, and I was in the lab actually getting the technology to work. And then I shifted to more computational approaches once we had all this data. And then when I graduated, went to Novartis, NIBR, so Novartis Institutes for Biomedical Research, when they first moved up into Cambridge, Massachusetts. And I worked there for about 14 years, doing computational research, and then for a while leading an experimental effort to do very large-scale transcriptional profiling. And I was really fascinated, or actually frustrated by, I knew how much data there was, in the public domain, and even in a big company like Novartis, certainly. But assembling it, when people would, for example, send us a compound that we profiled and we generate all this data, we'd have to go then assemble that data, even within our group, from multiple sources, and then around the organization and around the public domain. And it's just incredibly inefficient and labor intensive. So that's where I sort of had this idea, well, let's apply, what about Google? Google's done this for the web.
Yeah.
It can tell you what the web has to say, what the internet has to say about something. So took that idea and applied it to data, and we at the time implemented it and talked about it at meetings and things, disclosed kind of this approach, and sort of gave rise to this idea that, wow, we can really apply this to a larger scale as a solution to this data integration problem, right? And we realized right off the bat that it really, it checks a lot of boxes in terms of what you'd want from data integration, right? That it can handle this really diverse data, noisy data, give you results that are very concise because you get one sort of top hit, and has transparency because you can expose, here's the data that led to that top hit, here's the reason it's the top hit.
Right.
So that kind of gave rise to this idea.
Right.
And then ended up leaving Novartis, founding Plex, close to 8 years ago now. We're coming up on our 8-year anniversary. Back in 2017 that we started. And we built a system based on this idea of a knowledge graph. So kind of connecting data in a graph form where, sort of a network form, where anything can be connected to anything else, drugs connected to proteins and to diseases and the pathways and all of these different things. And then applying centrality algorithms to that. That was the core idea. And we've been using it ever since, applying it to all kinds of drug discovery problems. It's very general purpose. It's a very general solution. So many applications. And we've, at this point, worked with over 40 companies, biotech and pharma companies, on a really wide range of problems, and are just now starting to collaborate a bit more with academia. This year we have, or kind of starting from 2024, we have some academic collaborations that are starting to bear fruit in the form of publications, which we are kind of on the way, expect to have some of them out soon. So it's really been very successful, perhaps underappreciated, or we haven't sort of been loud enough about it, I guess, in terms of explaining to the world, why this is an important approach, showing it in action, and in ways that the public can really see. But we're starting to do that now. Yeah.
So we'll dive into the specific examples of what you've worked on in a second, but let's describe the scientific, the deep in the weeds scientific research that is actually would benefit from this. So maybe like, what are some teams doing now, and then what can, once instituted for Plex, what are they going to enable them to do?
Yeah, so there are a lot of different things you can do, which is generally, it's a strength, it's a good thing, but on the other side of that, I guess it makes it harder to explain and to cover everything. But to give some examples, there are a lot of efforts in pharma and biotech to do what's called phenotypic screening. So you'll run a large set of compounds and look for compounds that have an interesting biological effect. So kind of test them on cells or some complex system, and you'll have compounds that have this interesting activity that perhaps would be useful for treating disease, but typically we don't know how they work. We just know because the reason they're selected is because they do something interesting. Yeah. But the underlying mechanism isn't known right away, and in fact, it's pretty hard to figure out in general. It's hard to figure out what are they targeting these compounds and what's the mechanism. So one of the big use cases here is we can take these compounds, and just based on their chemical structure, we can sort of search all the world's chemistry databases and bioactivity databases, and say, well, compounds that have similar chemical structures are enriched for this kind of activity, hitting these kinds of targets or pathways or these certain mechanisms. So we've had a lot of success identifying the targets and mechanisms of small molecules, of these compounds, these chemicals. That's been one big use case. And then the one that we're really talking a lot about now and having, we've had a lot of success with this as well, is new target discovery and also biomarker discovery. So looking for, what's a new approach to a disease, what's maybe a new way to cure a disease, and identifying also, because we focus on the underlying mechanism. So along with that comes the underlying disease mechanism and the therapeutic mechanism, which also then leads to, well, here's the patients that would respond, right? Because there are often diseases can be complex, there could be different subtypes of diseases, there could be, genetic drivers where certain patients have the disease for a certain reason and therefore could be treated in one way that might be different from another set of patients. So because we sort of exploit or can discover the underlying mechanism, we can also understand, okay, what are the specific patients that will benefit? How do you identify them? So that's where the biomarker discovery comes in. So, so sort of the whole precision medicine package of here's the target, here's the patients that respond, here's why they respond, and here's the biomarker to find those patients.
Yeah, and just along all those elements of that journey, things are being just kind of vastly sped up with Plex. Just basically, you're getting that data and you're understanding.
Yeah, because we're identifying, we're using all of this data to identify the potential underlying mechanism, the underlying targets that are driving the disease and that could drive a therapy.
Yeah.
And again, the reason why it's we consider this discovery, it's really a discovery exercise rather than kind of just a predictive exercise. So we're not just making predictions about something. We're actually saying there's data out there that addresses this question that actually is informative. Yeah. Right. Even though the vast majority of this data actually is not in the text of papers or in the text of patents or the text anywhere. It's in these supplemental files, in these these large scale data sets that are collected, but barely, really barely utilized and barely explored. Yeah. So the answers to many of these things are actually in that data. And because we can surface it and we can ask across many data sets, where does that raw data, does it converge on something? Does it point to, a common mechanism? So we can approach the disease. Again, these high throughput methods have been applied in all kinds of ways. There are different, different data types. They're measuring levels of RNA and protein and different ways that genes are expressed, and they're looking at that at the level of the cell, at the level of organs, or they have these organoid sort of systems that are approximate organs, all the way up to animals and human clinical data. Yep. So there's just all kinds of data, and we can ask, if we look across all of that, is there something that emerges? Is there maybe a mechanism that we see commonly, whether we look at different systems, a disease model or the disease in humans, or if we look at the different levels of gene regulation, whether it's at the RNA level, the protein level, or genetics, right? If we look across all of that, does it agree on something? And by definition, it will agree on something. It'll be something that agrees on the best anyway. And we can then look at the top and say, often some of the things we already know will appear near the top, which ends up being a form of benchmarking. So we can see that this method actually can rediscover something that's known, which then can give more credence to other things that it finds. Because sometimes we'll even find things that have more evidence than the known thing, right? Or maybe just similar levels of evidence. So we can surface things that these insights and these potential these discoveries that have potentially substantial amounts of supporting data, even though they're entirely novel. They're things that maybe have never been described in the paper or patent, and yet we were surfacing this data that supports their role in this disease or in this therapeutic mechanism.
Totally. And are there data that do you feel confident in the overall data that you have access to, or are there data that's kind of spread out? Maybe it's IP of certain companies that you can't work with, and when you work with them, you can help them. Like, how do you think about that dynamic?
Yeah, I mean, there's just so much data in the public domain. We certainly can go. We also do work with companies, and we can integrate proprietary data in a very private, secure way just for them.
Yep.
But there's actually just in the public domain, there is just such an incredible amount of data that that doesn't hold us back at all. You know, so when we do projects that don't have access to any proprietary data or are just using public data, we can still surface all kinds of novelty because there's so much of it out there.
Yeah. Okay. And what has been the scientists and the researchers kind of initial thoughts of Plex? I'm sure they understand it, because one of your key insights was like, without this contextual data, the research isn't what it can be, and they understand the problem of it being parsed out and the research process. So just speak on their kind of initial thoughts on Plex once they see the demo or whatever.
Yeah, I mean, I think they certainly, when we present what we find, right? So we present the results. It's not any different from the kind of data they're already used to looking at.
Right.
So what we provide at the end of the day is a data package, right, of data points, and it's largely experimental data, almost exclusively experimental data that we found that supports the hypothesis that we're presenting, right, or the discovery that, the initial discovery that we're presenting. So sort of in that context or that framework, it's something they're used to looking at. They understand it. It's not any different from the data that they're used to dealing with. And that's something that's really in contrast to the sort of standard, it's become the standard AI methods, machine learning based methods, where they're predictions. You have to understand models and what was the model trained on and how was it validated and all of these things.
Right.
In this case, this is not an exercise of model building. This is an exercise of assembling data that already exists and looking for convergences of that data, places where that data agrees, or the consensus agrees on a result of interest. So it's a very different paradigm from prediction, from these machine learning approaches, and it's one that aligns, I think, in a lot of ways, it aligns much better with the scientific process, which is based on data, explicit data, and understanding the connection to that data and your conclusions. Because also with complex data, there's a complex relationship between that data and your conclusions, and you have to understand that. And so by providing the data we find, surfacing it in all of its detail, a scientist can really evaluate, here's what this really means, here's the confidence we can have in it, here's the places that it needs to be tested, where we would have further experimentation. It gives a tremendous amount of context for the findings, right, that you may not have with a predictive method.
Yep. Yeah, super important point. And you're working with biotech, pharma, academia, all of those kind of realms. Let's jump into the example that's in the actual paper itself on the Wnt pathway. So how does Plex kind of work with that?
Yeah, so for target discovery, there's a lot of different approaches that scientists use, and therefore that we can use with Plex to find new targets. One of those approaches is to look to use the idea we call phenocopying, where you can say, here's something I know, here's a biological effect that I understand, or I want to mimic, or I want to find something that reverses it. So, for example, some kind of disease state, and I can take a signature of that disease state, and I can go off and look for things that have a similar signature, or maybe reverse the signature. So, in this example, we look at a signaling pathway called the Wnt pathway. Right? So the Wnt pathway is important in development. It's important in some forms of cancer. And so we could take that pathway as an example. We could say, well, why don't it might be useful to find modulators, drugs, ultimately, that can modulate Wnt signaling, that can inhibit it, so the cells are no longer getting signal to divide, for example. And so a way we could do that, because we do understand something about the Wnt pathway, we can say, well, why don't we start off by taking known modulators of the pathway, right? So the ultimate exercise here is to find something new, but we'll start off with things that we know modulate the pathway. So we'll knock down certain genes, and there are certain genes that are known to be part of the Wnt pathway. Beta-catenin is one of them, ABCX and others. And so what we can do is we can start off, we can say, well, I know if I knock down beta-catenin, I modulate the Wnt pathway. So why don't I knock that down, and then I can go look to see, well, what other things are out there that kind of look like that. And in this case, when we say look like that, we mean, the genes that go up and down in response to beta-catenin knockdown. Is there something else that looks just like that, right?
Yeah.
That same genes go up and down, right?
Yeah.
So this is just one of many approaches, and this is the approach we talk about in the paper just as a starting point. We say, well, we're going to knock down beta. We're going to take data that already exists out there with beta-catenin knockdown and showing the genes that go up and down after you knock this gene down. So we take that public data set, that little sliver of data, and then we go look against all the other data in the world, and we say, is there something else out there where the similar genes are going up and down? And if we find something like that, then that would be something, another way to modulate the Wnt pathway, right? So we know beta-catenin modulates the Wnt pathway, and now we find other things that look very similar, so maybe they also modulate the Wnt pathway,
Right.
So that's the exercise that we do using just one sliver of data, one example of knocking down the Wnt pathway.
Yep.
And then we go out, and what we find is that, knockdown of other genes is very similar. And we find that robustly for a few of the genes, in that there's a very big overlap in the genes. Really the same genes are moving. And in one case, there's multiple members of a complex that no matter which one you knock down, the same genes move up and down. Right, so really high confidence that complex is involved. And if we if we just look at the genes that go up, we get an answer. And if we just look at the genes that go down, it turns out we get the same answer for some of these genes. So we see really reproducible, whether you look at the upregulated genes or the down. So it ends up being a form of reproducibility, right? A way to assess the strength of the connection. That we expect to see, no matter which member of the complex you knock down, you expect to see the same answer. If you look at genes that go up and genes that go down, you should have the same answer. If you look across different model systems and all these different things, if you keep seeing the same answer, you gain more and more confidence, right? Yeah. And so that's what we start to do in the paper. We only do really the first step. We do one data type, and we look just at one or two other data types. And we find some robust connections. And so that just illustrates sort of the methodology. Those targets could be interesting. They could be new Wnt pathway targets.
Yeah, are you going to further that kind of work?
So we are certainly furthering that kind of thing. So in the paper, we show kind of the first step of it, just to show the methodology. But the idea would be to take that approach and now scale it. Right. So what we showed in the paper, there were 4 searches that were run, for example, when we did this in the autonomous version. It goes and it decides, okay, we're going to take this approach. So the LLM, we actually have an integrated large language model that guides the searches. And it says, okay, we're going to take this strategy, we're going to run this search, and then we interpret it, and then we're going to run this next search and this next search. And so it does, it sort of does one or a few iterations of this, and ends up at some candidate new targets for the Wnt pathway. Right. And then the idea would be to extend that to say, okay, well, that's only a 10-minute run, a few searches were run. Just keep going and try modulating the pathway in different ways and looking for phenocopiers. Try it in different model systems. Try it with compound treatments instead of genetic knockdown. Try it in lots of different ways and see, tell me what's the answer, right, that keeps coming up. Like, what's the new target that we keep seeing, right, that no one appreciated that this new target actually modulates the Wnt pathway. But if we keep running these sort of virtual experiments, searches for data, if they keep coming back with, there's this gene X that whether we treat with a Wnt modulating compound or we treat with a knockdown of Wnt genes, and if we look at proteins, if we look at RNA, we look at genetics, we keep coming back to the same gene. Like, it keeps coming up, right? And whether we look in cells, or we look in organs, or we look in animals, or we look in humans, keep seeing the same answer, right? That's the kind of thing we're hunting for. And then at the end of that campaign, we would come back and say, okay, here's a slide deck, here's a summary of our research, right? That says, look, this target comes up, it's connected to the Wnt pathway in all these ways, right? In humans and mice and rats and cells and so on, and whether you look at RNA or protein or the genetics or other pharmacology, with compound treatments, here's all that data, right? And so you could potentially have that target could be entirely new, right? But still supported by potentially a really compelling amount of experimental data.
Right. Yeah, and so you mentioned LLM, so let's talk about the LLM piece and what exactly model you're using, which I believe is Claude at the moment, but that can change depending on the event of each one, and then how exactly you're getting the agentic approach to go on top of Plex as a layer, if you will.
Yeah, so we started out, we didn't foresee large language models coming along. So the technology before that was already very, very powerful. So you can run searches, you can find all kinds of things, and we have found all kinds of things. Now with large language models, you can tell them, okay, here's Plex, here's what Plex is about. We'll write sort of this prompt that gets fed in, before the user really starts interacting. We can give it background information and feed in this prompt that says, okay, here's how Plex works. Here's how to analyze the data that Plex produces. Here's the kinds of capabilities that Plex has. And that all gets fed in really as a prompt. So we're not doing any sort of fine tuning. We're not changing weights, anything like that. We're feeding in this prompt. And so now the user comes in and we have this large language model, and we're currently using Claude Sonnet 3.5, but we're ultimately agnostic about the large language model. So we can point to any model potentially. And so you come in and now you can start off and just say, well, just tell me about Plex. How does it work? And it can tell you about Plex. And you could ask it, how would I do X, Y, Z? And it can tutor you and describe how to use Plex. But then it can go beyond that too, because you can ask it, because it knows how to use Plex and it knows how to interpret the results. And it's really not so hard to get it to just actually run a search. So now you've got the entire capability is there for autonomous runs.
Right.
So you can go in now and say, I really want to find a new target in the Wnt pathway, to use our example. So we want to find a new drug for cancer. Right?
Yeah.
And you could just ask it, hey, how would I just do this for me, actually? So go ahead and plan a research campaign of searches and the kinds of searches that I was describing before. Just figure out what searches we would need to run, go run them, interpret the results, because it knows how to do that, and just do this on your own and come back to me later with all your runs and tell me what new target kept coming up, right? So what targets did you discover and what was the data behind it? So all the things that we just described a human doing as in a workflow, the large language model apparently can do this as well, can do it autonomously. So we can send it off and say, go do research, go find me a new cancer target. We have to, if we want to, get it to run in some reasonable amount of time, we probably want to specify what type of cancer, give it somewhat more information, but it doesn't really even need to be that much more narrow.
Yeah.
And it will go off and say, okay, I know the strategy to use, right? And I described one strategy, but there's multiple strategies. And it knows these strategies, and it'll start running searches consistent with those strategies and gathering results. And what it comes back with at the end is a data package, is a summary of, okay, I've run this many searches and analyzed them, and here's the strategy I used. So it's, the whole thing is transparent.
Yeah.
So you can evaluate, you can see the strategy that it did use, the data that it found, right? Everything is transparent.
Right.
And then it comes back and summarizes and says, okay, here's what I tried.
Yeah.
And here's why I tried it. And here's the data that I got. And here's the target I'm proposing and the data that supports that target. Right. So it really becomes it's a prototypical AI scientist. It can find new things. Right. And this is a real leap from what is out there. Right. Mm-hmm. And it was just last week that Google announced their AI co-scientist, for example, where it was about making suggestions about, okay, we've read the literature, and it's these LLMs have been trained on presumably mostly the literature and text-based sources, and it's proposing ideas, and it's all about kind of talking through these ideas based on the literature. Mm-hmm. So the novelty that you'll find just by sort of regurgitating literature, well, we don't know, and I'm not sure what we can really expect.
Right.
But if you instead point it at raw data that hasn't even been published before, and at least not in text of papers,
Yeah.
for the most part, or you point it at these large amounts of data and you do the kinds of things we're talking about, then instead of just suggestions about, based on reading papers, here's some suggestions, you can have actual discoveries, things that no one has described before.
Right.
But yet that have experimental support. Right?
Yeah.
There's data that supports these new
gathering the data in the corners of these repositories that are just lost around the world, instead of predicting something that may or may not be true, and it's on a scale of percentage basis, like you don't know, right?
Right. Yeah. So it has a bunch of features in that it's not predictive. We're not predicting something and giving some really abstract kind of statistical measure. What we're saying is that the data seems to support, this new discovery X, this new gene, let's say, this new target. And here's that data. Here's the data that we've uncovered that supports it.
Yeah.
Because the data was coming from all these supplementary files and databases, it could be that it was this particular, target hypothesis was no one ever came up with it.
Yeah.
And maybe no one was aware that there was so much data. Maybe they knew about a bit here and a bit there, but no one was aware of that. In fact, when you put it all together, there really is a compelling amount of data that's been assembled that supports this new target.
Yeah.
And that's what this system can do, is kind of assemble the full case from all of this disparate data and come up with new things that no one was aware of, that no one knew.
Yeah.
That goes beyond just a hypothesis. It's not just a suggestion. It's still ultimately a hypothesis.
Yeah.
But it's one that it's providing experimental data that validates to some extent.
Right.
And so, in a sense, it's really a new discovery. It's a new finding, and the data that supports it. There's always more to do in terms of validation, but it's providing you with a real advance.
Right. Yeah. All science is hypothesis, but there's hypotheses that are come to from predictive stuff, and there's ones that are come from real data, which is what you're providing, I think. So that's the key differentiator. And then in terms of the LLM, you're using Claude Sonnet. What do you envision, if anything, furthering the integration with Plex, making it better for users, or is now kind of just you're going to stick with what you have now and start getting to market easier?
Well, we're already using it. We're using it as an assistant, so you run a search and then it can help you interpret it. We're also using it in an autonomous way, and there's a lot of work to be done understanding the best ways to do that.
Gotcha.
There's this idea about what's the most effective way to chunk these projects, these efforts.
Right.
Do you have the large language model? Do you just say, okay, just cure cancer for me and go, and then it figures out all the agents it needs to create and all the things it needs to do? Or do you need to be more granular and say, I want, large language model, I want you to look at this from just the transcriptional point of view with genetics and just on this particular data set that I'm going to tell you about here. Yeah. And then here's another LLM, another kind of instance, another context where you say, I want you to look at this from a chemistry perspective.
Right.
And you go. Right. So we're still learning at what level this needs to be orchestrated. Can you just give it super high level guidance? I want to cure cancer, and then it figures it all out. Or do you need to be very specific and say, okay, you do the chemistry approach, you do the genetic approach, you do something else, and then be very explicit about how all that comes together. That's the kind of thing that we're exploring right now.
Yeah, and then tell me if this is right, will you be maybe possibly going into like letting the LLM understand or Plex understand the actual research process and like initiative that is going on at your clients, like in its entirety, instead of just like prompting it and giving it, hey, go do X, which is a little bit of one thing I need, without giving it the context of like X is going to contribute to Y, which is the whole process. So have you been thinking about that at all?
Yeah, I mean, that's really active investigation, active research.
Yeah.
We start off with trying to we try to be as general as we can, as general as the system can handle, and say, look, if you want to find a new drug target, here's an approach, or here's several approaches that we think will work, just in very general terms. Okay. In the hopes that then when the user gives it a specific and says like, this is the kind of cancer I want to cure, I want to investigate,
Yeah.
then it can generalize from our general instructions, and then it could give specific actions to the user.
I see.
But there may be cases where we may need to make it more specific for certain use cases, and give more details on a particular use case.
Yeah.
And all of that research too could all change. Like it could be that now the new version of Claude comes out, or OpenAI's version, GPT, or something else comes out, and now it is just smarter, for lack of a better word, and it can generalize more than the other model could. And so a lot of what we've done, where we thought, maybe originally we think we need to be very specific about how the whole thing is orchestrated. Maybe as the large language models get better, we have to do less of that, and the large language models can do more of it themselves, right? So I think that's going to be changing over time as the models get better, and we can just piggyback on that, because we're not training our own models. We're not, we don't really have a horse in the game, really, in terms of the models. So as the models get better, then this whole thing gets better.
Yeah, I think, and then eventually we'll get to a point where models are giving themselves context windows, and just not by a person, but as almost a different secondary pathway, just because they were told to do something and they realized they needed to. So we're gonna get to a point where they're actually training themselves, I think, at some point. So that'll be interesting. Let's talk about the Bird Institute, because you're helping academia as well. So I'd love to hear like working on the Bird Institute and how you're helping them.
Yeah, so we're working with Ann Carpenter's team at the Broad Institute on basically the cell painting data. So it's a technology that looks at uses what's called high content screening. So uses high throughput microscopy. So you're looking at cells, imaging cells, and they're stained in a certain way that's very uniform and high throughput. And you're looking at when I treat cells with a drug, how does that affect their morphology? When you look at them under a microscope after drug treatment, and you see these different kinds of cellular stains. And the idea is that if I treat two different cells with drugs that do something similar, then they'll look the cells will look similar, right? The morphology will be similar. And so it could be a way to identify how drugs work by matching them to drugs that have a known target. Right? So if you have an unknown one, and now you've got a similar morphology, then perhaps they both work in the same way. And you could do that with drug treatments, but you could also do it with genetic treatments, which is actually what the paper we worked on is actually on the genetic side. It's knocking down genes or overexpressing genes in this system, and then looking with the microscope at very high throughput, looking at all of these cells and saying, when I knock down this gene, the cells look the same as when I knock down this other gene, right? And maybe they look the same as when I overexpress some other gene. Right. So you can start matching up genes by the ones that give a similar morphology. They make the cell look a similar way, right? So it's a way to then use the cells and their morphology essentially to characterize gene function.
Can you describe morphology in more detail?
Yeah, so morphology, so the structure of the cell. So when you look at a cell, what's the size of the nucleus, and what's the shape of the cell, and things like that, that you would see under a microscope, especially after there's different stains that they use to stain the different parts of the cell. So they can look at, after I knock down a certain gene or I overexpress a certain gene, how does the actual morphology of the cell, the structure of the shape of the cell change? And if two different genes make the cell look similar, sort of change the cell in the same way, then it suggests that those two genes might have a similar function.
Right.
So it becomes an experimental way to characterize gene function, right? Kind of through the lens of cell morphology. Yeah. Right. And the thing is, so you can group these genes then. You can say, okay, now I've got a bunch of genes that seem to do the same thing in this context. And perhaps those genes weren't thought to do anything similar. Like they're not obviously related. So then the question would be, but they seem to be related because they have the same effect. Right. They seem to have the same effect. So this is where Plex can come in, and Plex can say, well, we can put those genes into Plex, into this sort of graph approach. This, we call it a focal graph approach, is how we characterize it. So you could take that list of genes and put that in our system and say, where have we ever seen these genes like together? right? So we certainly, now we've seen them together in a cell painting context, in this morphology, that they all induce the same morphology. So have we ever seen them all together doing something else the same?
Yeah.
And so we can go out there and say, oh, these genes have all been seen to change, the function of the mitochondria, for example, in the same way.
Right.
So perhaps the reason the cells look all the same is because they're all having a similar effect on the mitochondria, just as an example.
Yeah.
So what we did was we helped with that paper to characterize these genes that they identified, these sort of gene clusters, where a bunch of genes that all did the same thing to their cells.
Yeah.
We helped to characterize possible functions, like what is it that they could be all doing together that might have caused that. So that's the current work with the Brood. That's it's a preprint right now, and hopefully that will be published in the pretty near future.
Gotcha.
Yep.
And then, of course, University of Michigan, you're doing helping with drug-induced liver injuries. As well.
Yeah, so we work there with do a lot of work now with Johnny Sexton's group at the University of Michigan. They do a lot of work on liver biology. They specialize in approaches looking at drug-induced liver injury, different kinds of liver lesions like fatty liver, hepatic steatosis, these sort of things, and the underlying mechanisms and possible treatments. And so we're working on a few different things with them, and a lot of them are kind of in flight. But looking at, for example, compounds they identified that impact fatty liver, at least in mice, and seem to reverse fatty liver and maybe even liver fibrosis in mice, but have unknown mechanism. They've identified these compounds. They have these, they have the chemistry, the actual chemicals that maybe some form of them could be drugs one day. So they've identified them, but they don't know how they work, and so they came to us, and we put them in our system and actually have hypotheses about the potential targets of these compounds. And the first compound they gave us, actually, we immediately came up with, had a couple of target hypotheses, right? Basically, that we were able to say that compounds with similar structure are known to be inhibitors of the following targets, and it was actually DPP4, and which is a known diabetes target, and there's drugs on the market for that. And this other target called HSD, so involved in sterile biosynthesis. And that was, they presented that in a meeting in London, at the LREG meeting back in October 2024. And they're working on that paper. Still some more experiments to do to verify that. But actually, they did verify the inhibition of DPP4, which was one of the target hypotheses that came right out of our system.
Nice.
So so it's a nice example of we can, from structure, infer target. In that case, it was experimentally confirmed. We have other cases as well that that are proprietary that we can't really get into, but it's something that we see routinely with our corporate partners. But here's an academic group where the target is confirmed, and they're working on confirming the other target that the data points to.
Yeah.
So, and that could be, give some real insights into ways to treat fatty liver disease, which is a real need.
Yeah, and would they have confirmed it at all without Plex? And if so, how long? Like, what resources would they have at disposal to just pretend it didn't exist?
It's a very hard thing to do, to go from a compound to its target. And they, from what I understand, they ran the screen back in 2018, and they've been trying to figure out the target ever since, and hadn't. And we put it in our system, and we immediately ran it. We actually, in the introductory call when we first were introduced to them, we said just put the structure into the chat window. We put it into our system. DPP4 is the top 1 or 2 hit, and they hadn't seen DPP4 before come up. And we showed them, here's your compound. Here are similar compounds, structurally similar, and those structurally similar compounds are DPP4 inhibitors. That's what we were able to find very quickly. And that's the first thing they went and tested, and saw pretty quickly that, in fact, they are DPP4 inhibitors. So they had been, as I understand it, working on it since 2018, and with the right technology, we were able to immediately give them an answer that they confirmed soon after.
Okay. That's a potentially life-saving. I mean, the things you're working on, Aaron, it's it's not boring. You're fulfilling yourself.
Yeah. We take it for granted. I mean, I love to be in this field that the things when we make advances right there, in that case, where I think there's still more work to be done, even for that compound, because there's another target they're looking at as well that we predicted or we found evidence to support this other target as well. Mm-hmm. So if we can understand how those compounds work, that helps elucidate the disease process, identifies therapeutic interventions, and it could be this compound or something related to this compound, or it could be other things, because once we understand the mechanism, it opens up a much more rational drug discovery process. So the things that we find, especially because we're really getting, the goal, and often we're able to do this, is to get at the real underlying mechanism, like a true understanding of the disease process, right, and the therapeutic mechanism. So, we're curing diseases, or we're making progress towards curing diseases, and it's immensely gratifying.
And it compounds too. I mean, once Plex finds a commonality and identifies something and confirms something, that goes into, I guess, the world's database as well, which then can benefit the next Plex or Plex itself working on a similar idea. So,
yeah, like it's very durable because we're getting at mechanism, so we're getting at an actual understanding of what's happening. So that's real knowledge. That's durable knowledge. Yeah. We're arriving there using explicit data that we've uncovered, right? So we also know how we got there, which is important. So that also makes it durable, and we have some confidence in it because we know how we got there.
Yep. That's a good question. Are you for the customers or the work internally that you're doing, where you're confirming these findings, are you putting them into some sort? Are you putting them to Plex itself as well? Like, you know what I mean by that? Are you actually like cataloging the findings and confirmation that Plex is aiding with, I guess?
Yeah, I mean, we can continue the investigation, right? There's always more to do, either in the lab, going to lab, but also just going back to Plex, because there's just so much data and so many different approaches you can take. So you can say, okay, we're now confident, or we've made some progress in understanding the mechanism behind fatty liver. Yeah. Right. And so now, and we think it has to do perhaps with these couple genes. Yeah. And so why don't we now come back and we say, well, let's explore these genes, and is there evidence that these genes impact fatty liver in other sources, in other model systems, and other data sets, right? So now, instead of coming in with chemical structure, which is how we started, now you're coming in at the gene level, and you're saying, how about if I look at knockdowns of these genes or overexpression, and we look at different kinds of systems, including model systems that are relevant to fatty liver, are we going to get more detail about the underlying mechanism that might be really useful?
Yeah. And then as data gets more complex from research, how do you foresee Plex kind of making sure it's staying on its game to get because it's not going to stop. I think it'll get more complex, and it might be more organized thanks to AI, but it'll still go. We're still going to do research, obviously.
Yeah, I mean, it's been complex, and it's not going to get any less complex. And so this is a way to say, to at least use the data. I mean, it has to, at the end of the day, however complex it is, someone's got to understand it. I mean, generally there's a paper, there's scientists who designed it and are interpreting it. Yeah, right. So there is some way to interpret this data. It can be that each data set needs a different kind of interpretation, and that's more or less how things seem to go. But what we can do is we can capture each of those interpretations individually. We can say, okay, here's this new data set's been generated. Here's the proper interpretation. Maybe it connects genes to diseases. So we'll connect genes and diseases using that data, and that goes into the system. And we can apply whatever the proper interpretation is for each data set. We could do it data set by data set. And each time we do that, it accumulates. We put it in the system, and now that has accumulated knowledge of the system. So now when you ask it a question, a new question, or maybe the same question you asked before, but now there's more data. If that data has something to say about it, now it comes up. Right. So it could be before, you asked that question and there was an answer, but it only had one data point or a couple data points to support it. But now you keep adding data, and now you go ask the same question, and now the answer, maybe it's the same answer, maybe it's a different answer. but it now has a lot more supporting data than your old answer did, right? So the more data that goes in, the better the answers get. But it remains concise, because there's still going to be one winner. There's still one thing that's the best answer, at least that has the most data, and there'll be a second best answer. So no matter how much data goes into the system, it doesn't lose that concision. It always has that winner at the top, right?
Yeah.
And it maintains the transparency, so you still know why it's the top hit, why it's the most compelling answer. And so and that itself is really an important property. So it means we don't have to worry about drowning in data. We could just keep piling, as long as it's reasonably good data. Doesn't have to be perfect data, right? Because again, it's a consensus method. So if it's really noisy data, it just won't agree with other data sets, and it just falls to the bottom. So we don't have to be overly worried about noise.
Right.
So we can just put in, our best shot at these data sets and the best interpretations of these data sets. They go in, we accumulate and accumulate, and the more that goes in there, the better these answers get, right? There's also this opportunity to, there'll be maybe a way to measure, on average, for some, if you just randomly choose a disease, there's on average the best target that we've come up with has a certain amount of data, right? And the more data you put in the system, the better your average disease query will get, a certain number of data points supporting it, right? But you can also then go on sort of look at the extreme. You could say, well, that's fine, but what's the disease that has the best answer?
Yeah.
Right. So what if I just want to know, what's the best opportunity out there, right? Where have we convincingly identified a new drug target, a new way to cure a disease that we didn't know before?
Right.
Where there's the biggest delta, where the data that we didn't know about is really just pretty massive.
Yep.
So you go look out on the tail, and you can just have it run a lot of searches, right? And you can say, actually, here's the place where there's the biggest delta, where it can find the most information beyond what we already knew. Yeah. Right. And you can even combine it then with other ways to define opportunity. So I want not just a lot of data. I also want it to point to an already approved drug, right, that everyone could just go and take tomorrow, right? Because it's maybe even over the counter or whatever it is, right? So something that's very actionable, something that's could have a lot of impact. So you could take the whole system, especially when you're running it at scale, and be very opportunistic and say, okay, go out and find the biggest opportunity, and then define opportunity by really compelling data, availability of drug, maybe a repurposing opportunity, however you want to define it. Right?
Yeah. Is that how you see? So, I mean, that's my next kind of segue question is, how do you see Plex? Maybe they're one and the same, but how do you see Plex and how do you see drug discovery in the next, we'll say, 4 to 5 years?
Well, I think what this exposes is that we know a lot more than we think.
Yeah.
So, and I think this is really important, right? There's a lot of cases where we think we don't know how to cure the disease. We have all these rare diseases, for example, that we think we don't know very much about it. But in fact, in many cases, we may know a lot. Right. Because that gene, let's say that maybe the gene is known for the rare disease. We know the gene that causes it. And we think, well, but we don't know a lot about the mechanism or whatever it might be. But that gene has been measured all over the place.
Nature nature has the answers. We just need to kind of go through the giant storybook of nature and figure out what those answers are. Is what you're saying. Right.
Yeah, so that gene has been measured in all kinds of contexts, right? Yeah. Where people weren't thinking about this rare disease. They were just measuring all the genes because that's what we do now.
Yep.
Right? So it's been measured all over the place. And some of those measurements are relevant to this disease. So we can go out and find those and we can say, actually, we do understand this, right? And often the mechanism, like you said about biology, biology there's a lot of conservation through evolution. That even though, we may think, okay, this is a rare disease, this is a gene that, very few people have mutated, and, even so, it still could be related to other processes that we do understand. Right. Because there's that level of conservation. So it could be that, yes, that gene, very few people have that particular gene mutated, but that gene participates in a process that is conserved. Yeah. Right. That maybe we've actually studied, knowingly or not, in other contexts, where that's conserved in other animals or other related processes. So often, even though we think that it's very unique and there's nothing else like it, because of the conservation biology, there's often a lot more information out there than we realize. And that's one thing that we can uncover here.
Totally. Yeah. Where would you like to look back, maybe if you could look down on Earth in 100 years from now and see where Plex is? What would you like Plex to have accomplished?
I think ultimately it's about understanding. So I think that because I think a lot of the things that we're trying to solve, we have all these observational studies where we find these correlations between things, and which can sort of give us hints about things. But until we really understand the underlying mechanisms, it's not solved. And I think that we have a lot of the kinds of experiments we're running these days can get at mechanism. They sort of are approaching mechanism for all these different angles. And if we pull it together, we can really understand that the underlying mechanism. And then things get a lot more definitive, right? Because I think you see right now, because we have all these imperfect methods, a lot of correlative kind of methods, and that we see kind of this swinging back and forth about, this is good for you, this is bad for you. There's just so much uncertainty that I think if we understand underlying mechanism, a lot of that disappears. And we say, no, actually, this is what's happening. And we know because we can tweak the different parts of it. We know exactly what's going to happen because we understand it.
Right.
And we know that, if you give this to a different person with a different genetic background, this is how they're going to respond because we know mechanistically the difference between these two people, right? So I think I'm hoping, sometime in the future, we'll look back and say, okay, back in, 2025, when we didn't understand all these mechanisms, we were groping in the dark, but now we understand how this works. And so now we know what's going on, and we know how to confidently manipulate the system to, cure disease or prevent disease. Because we know why it's happening.
Yeah, great description of like what you're trying to accomplish, I think, from that answer. And it's just kind of so you just want to aid human knowledge to understand definitively mechanisms going forward in time, and that's something that'll make you happy.
Yeah, I think we need to be focused on mechanism. That's where it's not solved until we understand the underlying mechanism.
Yeah.
And I think that the contribution of this sort of approach is that we've collected incredible amounts of data and a lot of which we just don't know how to use, right? And there's been for decades now in this field a lot of hand-wringing about silos. So we're certainly not the first ones to say, oh, the data's in silos. Everyone has been wringing their hands about that for quite a while now. But I think what we need is a method that can break those silos, that can actually use all of this data, right? And I see, people in this field, and up to, I think even today, was it today or very recently, there was another Nature opinion about we need better data, cleaner data, some aspect of that. It's been always the same complaint. It's been, the biologists need to go to the bench and they need to create cleaner data, better annotated data, the right kind of data, all these things so that we can train models and they'll work, right?
Yeah.
What we're saying, and that I'm sure that's true. I mean, models need that kind of data if you're going to get models to work, at least if you're get them to work on everything. But what we're saying is that there's a different approach, right? It's not instead of model building. It can be very, very, very synergistic with model building. But there's this other approach that works just fine with siloed data, right? It works just fine with noisy data because it's consensus based. So we can use it with the data that we have. We don't need to be, harassing the bench scientists to more data, better data, annotate your data better, all of these things. You could say with the data we have in the state that it's in now, it's already tremendously useful with this approach, this sort of search-based approach. That this algorithm, this approach can use the data we have, right, and can make progress with what we have. And I think that's underappreciated. Right? There's still the hand wringing that like, oh, now to enter the age of AI, we've got to clean up all our data and find all this, and that can't hurt. But we have a method now that can use the data we've got, and we should be using it.
Yeah, awesome. Do you want to share like what's next for Plex, what you're working on near term?
Yeah, so we're, I think we're out there. We're trying to communicate this. So, and this is why I don't know if I said it before, but I appreciate the invite to be on your podcast, to be here and to be talking about this.
Yep.
We're we're doing the work. So we're working with biotech and pharma. Mm-hmm. We're also working with academia and government as well, research institutes to make progress. So we're doing that, and we're really excited about that. And we're trying to get the word out because there's tremendous excitement about AI, right? But essentially all AI has been machine learning based. You can argue that what we're doing is an agentic approach, which it certainly is. So this is an agent, right, that can work together with large language models and other kinds of machine learning. And it's sort of a unique agent, right? So we've seen the power you get from combining large language model approaches, for example, with internet search. That's been maybe the most common tool to link to it. Yeah. And it's tremendously powerful to do that, right? And I think at first it was called RAG, or retrieval augmented generation. People are talking about agentic approaches where you have tool use, where maybe an internet search is a kind of tool. So this is an agent, and it's an especially impactful agent because it can take biomedical data, and again, to kind of repeat a bit, not just the papers, not just like what people wrote in the papers, which is the tiniest fraction of it, right? But actually the relatively raw, genes going up and down, and the structures of molecules and their activities and all these things. It can take the raw data and tell you, okay, here's the answer that it all seems to be pointing to. And so you link that up to a large language model, and you ask it things like we do, like, well, give me a new cure for cancer, targeting the Wnt pathway. And it goes out and it actually can come back with things that are in our data that we didn't know. And, so I think back to your question, so what are we trying to do? So we are certainly having the impact in these companies and in academia and these research institutes. Yeah. But we really want to shift the conversation or add something to the conversation that we can actually make a lot more progress than we're making, that we're actually not very far off from AI scientists that we can, they're not entirely autonomous now. We need to supervise them and guide them and refine them and do all these things. And for the foreseeable future, there's going to be a need for scientists in the loop and scientists guiding the process and evaluating the process. Yeah. But these agents can work in a much more autonomous way than we appreciated. They can find really new things, and that's been a debate, right? And even as recently as this last week or so, with the Google AI co-scientists, a debate of, is it really discovering new things? That's really debatable. I think with this approach, it's no longer debatable. It's right. And it's not that we've discovered a target that's now in clinical trials, but we've discovered targets, at least on the first steps, where we've discovered targets, we've shown the data that sort of supports that discovery, and we've done it by a process that's very clearly scalable, that could clearly find more validation data. And although we haven't gone sort of all the way yet, it's clear that running this should result in targets that potentially have very compelling amounts of validation data, despite them being entirely novel. We should be able to surface experimental data that supports them. So I think we've shown that all the pieces are in place to have a largely autonomous AI agent that actually makes new discoveries. And I think that's a really big deal.
Yeah, it's going to be an exciting 2025 for you.
Yeah, I think so.
Yeah, excited to see what's next. Doug, thank you so much. Great.
Thank you, Taylor. Thanks for the invite.